Cool new technology. Fun, fun. Hopefully this will work. We shall see. I don't know if it's actually going to get recorded or not, but I'll give it a try. I did upload first lecture onto YouTube. Hopefully people have found that. If you want to subscribe to my YouTube channel, go ahead. I don't care if you do or not. It just tells you every time I've uploaded a new lecture. So hopefully all that will work. People who've had my classes before, I know there are a couple of you, know that usually once or twice a term technology doesn't work for whatever reason or other. Speaking of technology not working yet, our favorite thing up here, clicker questions. I tried to upload them yesterday and figured that out, and they changed the system on me, so I'm still working on that. But just in order that we get used to using our clickers, we'll have another clicker question, which should be really straightforward and easy for people to deal with. So again, practice-wise, I'd like you to answer the question without talking to anybody. If everyone gets it right, then we'll move on. If not, then we'll have a chance for everybody to chat with each other and then vote again. It's the last vote that actually counts. So there is the most molecular diversity in which of the following? I don't need this anymore. Archaea, animals, plants, fungi, or humans? And let's go. And yes, clicker questions are not going to actually start counting until next week. So give people a chance to get their clickers and make sure all the registration works, etc. Although registration is not a problem because I have all of the scores already. It's just connecting your clicker number to your Odin username and making sure that those connect. That's the only issue there. So we've got 100 and some people with clickers today. That's better than on Monday, where I think we had 60 or so. So we're getting there. Hopefully, again, I'll be able to find all of these, and I'll just upload what's happened for today when I get a chance to do that. Okay, let's see how people did. It looks like, well, we can look at our responses here, show our results. Again, if it had been less than this, then I'd let everybody chat. Those of you who had B, C, or E are either messing with me or it's something that we could chat about. But yeah, yes, Archaea. Why is that? It's because it's the largest group of different organisms. In fact, there's way more diversity in Archaea than animals, or for that matter, probably even the crown group of eukaryotes at a molecular level. So again, the key here is what's the molecular diversity, which is present in these different organisms. So let's see, is this going to work? I'll hide these quickly, and I will actually select this as being the correct answer, so you should be able to see it when I get around to uploading these scores. Okay, so we talked a little bit about this last time. Just really quickly in review, where do new genes come from? Last time we talked about the last universal cellular ancestor, which had probably between 300 and 500 genes. We've got 20 to 30,000. Where did all of those extra ones come from in the intervening 3+ billion years or so? Some of it came from the just mutations that happened in normal genes, but then gene duplication is a major part of going from one to many, and we'll see this happens very frequently. As mentioned last time, since you have whole genome duplication. Once you have gene duplication, then you can mess with one copy and have the other copy either stay the same or be variable. But probably the best way, again in my slightly biased point of view, is to have horizontal gene transfer, which is a way that you can have an organism, in this case the organism B, pick up genes from organism A through this process called horizontal transfer. So where we stopped last time was where, See, my technology is not working the way I'd like it to. How annoying. Let's see, can we even advance the slide? There we go. Homology versus what people usually call homology. So lots of people talk about homology. Whenever you see homology in the literature, far too often it's sequence similarity. Well, sequence similarity is really not the definition of homology. Homology means if you've got two homologous genes or homologous proteins, that means they've come from a common ancestor. They may or may not have detectable sequence similarity with each other, and you can have sequence similarity in the absence of having something which is a real homolog. So this is, I will probably try and repeat this many, many times throughout the rest of the course, but homologs mean common ancestry. They don't mean a similar sequence. And this bugs me all the time in even the scientific literature. I'll see something that says, these genes are homologous to each other. No, they're similar to each other. You don't actually know if they're homologous. They probably are if they have similar sequences because they've probably descended from a certain or ancestral organism, or actually even more importantly, an ancestral gene. So that's the first concept, is homology. The second one is the one between paralogs and orthologs. You know, what the heck's a paralog and ortholog? Those of you at organic chemistry are actually have a step up on this one. You know, para versus ortho in terms of where you have your substitutions on the benzene ring. But the basic message here, at least as far as we're concerned, any two genes that are different from each other because of a speciation event are going to be orthologs. So you had a gene here. You have two different species. You have a gene now which is homologous in these two, gene GB. These are homologs of each other. And they're orthologs because they're present in different organisms. The flip side of that is a paralog. And the paralogs are when you have gene duplication. Oops, let's see, I do this way over here. Still getting used to this thing. Here we are. Gene duplication in a single species. And then you'll have mutation, you'll have rearrangement, you'll have something else. And so G1 and G2 now are paralogs of each other, whereas GA and GB were orthologs relative to each other. And paralogous genes, orthologous genes, these are actually really pretty important because if you think about, again, the expansion of the number of genes, that's always going to be paralogs. And the great thing about paralogs is that you've got a backup copy of one of them. So you can play around with these paralogous genes, and often they'll have slightly different functions. On the other hand, orthologs, you've only got one copy of it. So it's probably just going to have very, very similar functions to each other. So why do I spend so much time talking about this? It's because in many cases, we have what we call gene families. And the book spends a whole, I think, if anything, far too much time talking about gene families. But the basic idea here is if you have a whole bunch of homologous genes, i.e. they've come from a common ancestor, they're going to be a gene family, and they'll have various variations that you have between them. And so what you find pretty typically is a situation not unlike what we have here. So tubulins, if you just look at tubulins, tubulins are really important for cell structure. We've got 8 different tubulins. We've got alpha tubulins and beta tubulins. Alphas and betas are paralogs relative to each other. So if they're paralogs, they're likely to be what in terms of their function? Identical or different? Where's my name? So actually the opposite. It's usually they'll be different. A paralog, you've had 2 genes that are now in the same organism. So often they're going to be a little bit different in terms of their function. There was gene duplication that happened in one organism, and then they've diverged. On the other hand, the orthologous genes, in this case, for instance, alpha tubulin in humans and beta tubulin in humans, these are the paralogs, but alpha tubulin in humans, alpha tubulins in flies, these are now going to be orthologous to each other, and they're likely to have a really similar function. They were now orthologs, are different species, but there was that one gene that had that one activity. And so those are likely to be maintained. And it's in, you've got orthologs and paralogs they come together to make up these gene families. And gene families, they're basically going to be homologous genes. I'm going to have similar functions, but again, the paralogs are often going to have more different functions. Orthologs are much more likely to have very, very similar functions. And so this is how we generate lots of gene families. And see in just a second when we look at a table, and as you download the notes, we'll have seen it already, that we have many, many, many gene families present, particularly in our genomes. So yes, we have way more genes than E. coli, but often there'll be one gene family that you have in E. coli, or at least in some other model organisms, but it's just been expanded a whole bunch in the human genome. Other question, where do you get lateral gene transfer from? Again, I like this particularly, but it really gives you a way that you can add genes to one genome much more rapidly than gene duplication or excuse me, whole genome duplication. So lateral gene transfer, all that is, one gene moving from one organism to another organism. The best way that we know that this happens has to do with my favorite set of organisms. Again, are they alive or not? We can discuss that in much more detail. But viruses, and particularly true in bacteria. The transfer of genetic material through viruses from one organism and literally one lineage versus a different lineage, that horizontal gene transfer is rampant today, was probably even more rampant back when life was originating. Another way you can think about lateral gene transfer is the evolution of sex and sexual reproduction, because if you think about it, you've got two organisms, and they're cutting out half of their genes in most cases, and then only transferring half of them. So you can also have pretty major changes at an evolutionary timescale in terms of genes through this sexual reproduction process. So lateral gene transfer and then gene duplication. That process generates gene families, and these are some of those gene families. And particularly important here are those gene families which are conserved in all organisms, in this case, all three domains of, yes, it's got the living world. That's because these are people who are cellular biologists, not real biologists like me. No, these are in cellular biology. These are gene families that are present almost everywhere. And the main one to mention here is translation. 63 gene families that are involved in translation that are found in every single domain. And if it's found in every single domain, what does that mean about its function? Probably going to be really, really important. And all of you remember from that minimal genome that we talked about on Monday, what were those genes that were actually still in that genome? Translation genes. Some metabolism genes too, but mostly translation genes. So translation, really, really critical. And probably that was one of the first things that arose way back when in that last universal common ancestor. What's the gene that we use to make those massive trees that have all these three different domains? See, I really do need my list. The 16S or 18S ribosomal RNA gene. What's the ribosome? What does it do? Translation proteins. So yes, translation, really, really fundamental, and again, probably absolutely critical for cellular life. Everybody's got it now. It's all similar in each of those different organisms. So these are the gene families. Now we get to the question about, well, what do genes actually do? And how do you figure it out, much more importantly? And a couple of people asked me about this on email. Now, is this year going to be similar to last year? Yes, in terms of the syllabus, with a couple of differences having to do with methods, because I think we should talk about methods earlier on in the class than the textbook does. So that's Chapter 8, which we'll be doing some of in the lecture on Friday. So how do you figure out what a gene does? And you being biologists, people are looking at this. I kind of see three main ways of doing this. The first one is genetics. And what I like to say about geneticists, what do? They break stuff and see what happens. So you have a mutant. You figure out what the phenotype is of that mutant, and then you try and figure out, going backwards, what that means. An incredibly powerful technique. There are some issues with it, and the major one is that most mutants kill off whatever you're trying to work with. So that's a bit of an issue. The opposite end of the spectrum, sorry, back up a little bit. The idea of a genetic system, of course, is you've knocked out one gene, or maybe a couple of genes, in an organism in the context of all of the other genes which are there. And so that gives you a pretty good indication where that gene and the function of that gene fits into the whole rest of the organism. The flip side is to do biochemistry, which is very much the reductionist approach, where you now take one particular gene, and very often that one gene product, and then figure out what it does in the absence of everything else. And that will give you an idea how that's likely to function. Enzymes are really good examples of this. So you have a gene for an enzyme, you make that enzyme, and we'll talk about how you do that in the next couple of lectures, and then you use an activity, you figure out what the activity of that particular enzyme is, and say, oh, it's probably likely to be phosphorylating glucose, hexokinase, or something like that. So, and then you can say, oh, well, what is that likely to be doing then inside the context of a cell? And the really powerful experiments actually then going to be combinations of the two. So you do a genetic experiment, you think you know what that gene is doing and how it's important. Then you take the product of that gene, you study it in isolation, and you see what it actually does in isolation. If you can really put those two together, that really gives you very powerful indications of what a particular gene, and particularly a gene product, is actually doing. There's another way, I think of this as kind of the lazy way to do it these days, down at the bottom, the bioinformatic technique. And basically what that is, find a similar, hopefully homologous, not always homologous, gene that somebody else has done all this work for. And there have been enough molecular biologists, cell biologists, geneticists working that very often when you go out and find your new favorite gene in your new favorite organism, somebody has found something similar already. And the power of this bioinformatic approach is also incredibly useful and allows you to at least make predictions in terms of what you think is likely to happen with that particular gene. And then you can do the genetics, then you can do the biochemistry if you want to figure out the rest of it. So any questions at this point? Yeah?
Can you clarify ortho and paralogs again? I wasn't exactly clear on what you meant. Where, like a definition and then the question you asked, I got a little confused.
Okay, yeah, so the definition of orthologs versus paralogs. Should I back up to that slide maybe? Maybe get a little bit easier? Or, okay, just to walk through it. So orthologs are going to be two similar genes which should be homologs of each other. So the question is, are they orthologs or are they paralogs? They're orthologs if they're homologous genes which are present in different organisms. And so like, human and yeast, for instance. Now, if you have a paralog, those are two homologous genes, but they're present in a single organism. And so the example we had there for tubulin, you have alpha tubulin and beta tubulin in humans.
And they're not identical.
Those are not, none of these are identical to each other. They're going to be homologous to each other.
Which is common ancestry.
Which is going to be a common ancestry, exactly. Thank you. Other questions? Paralogs, orthologs, homologs, gene families? Everybody's totally clear on this, which means they'll be able to answer a clicker question on it. Yay. Okay, so which of the following is the best description of genes that are in a gene family? They're homologs, they're paralogs, they're orthologs, they're paradoxes, they have identical sequences. And I hope everybody picks D. And again, take your choice. We'll see what the answers are, and then we can go from there. Okay, we've got about 70% of you who are thinking it's one answer and about 30 who have less than that. So go ahead and talk to your neighbors. Tell them what you decided, and then we'll go ahead and answer it again. Go for it. Talk to your friends, neighbors. Who are they? Why did you select what you did?
So it tells me to hold it until the blue light flashes.
Yeah.
Then your frequency is AA.
That's correct, yes.
So I click AA.
Yep.
But how would you know that it's me?
You should register it when I upload the scores, then you should see it. If you don't, let me know. But have you registered it yet? Have you registered that clicker with that number? No, you need to do that first, because otherwise I'm not. But I have the clicker. I'll have that clicker number, which shows up here.
Oh, okay.
Yeah. I just need to make sure it connects to you. Okay, we ready to go again? Everybody happy? Yes? No? More so? Go. And for those of you who are not used to the clickers, it's your last push that actually counts. So you can, unlike in other elections, vote early, vote often, but it's still the last one that counts. Okay, so nobody liked paradoxes. They don't have identical sequences. That was a good question as well that you led into there. They could be paralogs or orthologs. So what does that leave you with? Homologs, exactly. So hopefully this is useful and that discussion is useful. Definitely, it went from 72% to 84%. So hopefully there's some advantage with that. Okay, so those are your gene families. A lot of the fact of your gene families then is what gives us different genome sizes, is the presence of all of these different gene families, and particularly the size of any particular gene families. Some gene families, I think the textbook is the example of the Sark gene family, has literally hundreds of members in that particular family. So many, many, many similar kinds of proteins, and again, the homologs. But they're going to have, depending on if they're orthologs or paralogs, potentially slightly different functions relative to each other. But the big difference is in terms of genome sizes here, and this is a log scale at the bottom to remind you of that. We can go from the smallest genomes, and this of course is out of date because I showed you a genome on Monday, which has literally under 200,000 base pairs inside of its genome. But the largest genomes are actually, that we know of now, are the protozoans. Some amoeba have these genomes which are hundreds to thousands of times bigger than our genome. Why is that? Nobody really knows. And what are all of those extra pieces of DNA? Most of them are copied pieces of DNA. Some of them are going to be families. A lot of it's actually non-coding, which is kind of interesting. And we'll look more at the human genome when we look at this later on. If you just look at the human genome, only 1.5% of the human genome is actually protein coding for unique genes. Actually, a bunch of duplicated genes that we'll talk about again later. But relatively small amount of non-protein coding. Is the rest of it junk? That's a whole big discussion that molecular biologists have been having for a few years. The answer is probably not. There's probably functional, but what that function actually is very challenging to figure out, because mostly you can't really do biochemistry on a piece of DNA that you have no idea what it's doing. and the genetics are much more challenging. Yeah, you had a question over there?
Yeah, I was just wondering, is genome size independent of diversity in function or molecular diversity?
So the question is, I guess just to repeat it here, is genome size independent of molecular diversity? That really depends on how you define molecular diversity. It is true that the vast majority of these really big genomes are duplicated DNA sequences. And so in terms of actual complexity, in terms of different sequences, they're probably not that different. But again, it really depends on how you're going to define complexity. But yeah, a lot of the big differences here are actually differences in repeated DNA. One of the reasons that the plant sequences are often very, very large is that some of them are tetraploid or octoploid. So instead of being haploid, 2 copies of the genome, they'll have 4 copies of their genome, 8 copies of the genome. And so that immediately is going to make you much, much larger. So, but quite why that is, again, never ask why questions in biology. The answer is always evolution, which is completely obfuscating because it's not really a good answer either. It works. And again, why amoeba and other protozoas have these massive genomes? Some people think it's because how do amoeba eat? They eat by engulfing things. And maybe they just pick up a whole bunch of DNA from the things that they've engulfed. Possibly. Again, open question. But what it basically says is there's lots of possibilities. And size doesn't matter, at least in terms of genome size. So those are, just wanted to mention that. The other thing is one of the reasons that I like to study archaea down here at the bottom is because they actually do have relatively small genomes. And figuring out what each of those genomes do, or the pieces of the genome do, is more straightforward than something that's got a whole bunch of non-coding sequences in it. We're going to talk a lot about E. coli in this course because it only has 11% of non-protein coding genes. Makes it a lot easier to study. Doing genetics is easier. Doing biochemistry is easier with these. So that brings me to our model organisms. It would be great if we could study absolutely every single organism that was out there, and people are now trying to do this because sequencing has become so relatively easy and relatively cheap. But be that as it may, since we looked at all those gene families, everything's descended from a common ancestor. If it's true for E. coli, it's true for the elephant. It makes a lot of sense to study organisms that are easy to study. And one of the easiest ones to study is E. coli. A couple of reasons that it's easy to study. One of them is that it's a relatively small genome. Again, lots of coding sequences, 4.6 million base pairs as opposed to the human genome. Haploid copy 3.2 billion base pairs, and around 4,000 protein coding genes. These numbers, so when you see genes, the number of genes, this is actually kind of flexible. People are finding new genes in different organisms all the time. We talk about genes if it's a protein coding gene or a non-protein coding gene, a gene which codes for an RNA. It could be a functional RNA. It could be a regulatory RNA. So we really still have a great handle on exactly what's going on here. But it's a nice system to study, really easy to grow. It's got a lot of the fundamental gene families, again, for transcription, translation, etc. And when we talk about something, and particularly this textbook, it works this way in bacteria. 99% of the time it will mean it actually works this way in E. coli. And I could have just as easily had bacteria as the first answer for that first clicker question. Way more diversity in bacteria than there are in animals, crown eukaryotes, etc. So when we say bacteria, it usually means what's happening in E. coli, and there are some definite exceptions to that as well. I like to think of Sulfolobus sulfotericus, my favorite archaeal organism because our viruses grow in them, as also being a nice model organism. It's easy to grow despite the fact that it grows in boiling acid, and relatively small genome and relatively few number of genes. So we like to use that one. In eukaryotic systems, not surprisingly, we care a lot about us, understandably, how we work, particularly from a medical point of view. But humans are really hard to work with. They don't mate with the right people that you want them to mate with, would be convenient if you're trying to do genetics. And their genome is ridiculously big. They live for too long. They're hard to grow, billions of them in small amounts of spaces. So people have used some other organisms to try and understand some of the more fundamental aspects of how eukaryotes work. The first of those to be really studied in detail, kind of the E. coli of eukaryotes, is Saccharomyces cerevisiae. The brewer's yeast, again, really easy to grow, relatively small genome, relatively few number of genes, but it's a yeast. So if you want to understand something about nervous systems or vertebrates or physiology, it's kind of hard to study yeast. So other people have figured out other organisms to study. C. elegans, Cenobambatus elegans, is a really fascinating organism that many people study, including Dr. Estes here on campus in the biology department. These are small worms, and they're really neat to work with because they're completely transparent. And you can see all of the cells as the cells divide. They also have a nervous system which is reasonably well defined, but really from a cell biology and developmental biology, it's really nice to work with. Under 1,000 cells, and people know exactly where each of those cells came from. I should have brought the issue of Nature, not Nature, Science, from their breakthrough of the year last year. It was talking about cell lineage and exactly where cells come from, when they undergo development, and what genes get turned on at one particular time. And so this is the big breakthrough of the year. You can actually study individual cells and what genes get turned on as each of those cells turns into a different cell as it's undergoing differentiation. Arabidopsis is the model plant organism. If you remember our figure, I don't know if it's back here, here we are, of the different plants. If you look at Arabidopsis, it's a relatively small plant genome, hasn't undergone all of these extra gene duplications, also grows pretty fast. It's a weed, and you can do pretty good genetics with it. Drosophila, also known as the fruit fly. I certainly have a lot of them flying around my compost, so easy to get lots and lots of them. The genetics are pretty good. This was really developed at Columbia, kind of the turn of the century, looking at a genetic system. A lot of what we know about development from a molecular point of view comes from this original genetics. And we'll talk quite a bit about genetics of fruit flies and fruit fly development. It turns out that development in fruit flies, even though they're invertebrates, turns out to be very similar to the way a lot of vertebrate development works. If, however, you really want to work with a vertebrate, fish are a pretty classic way to go. We actually have a whole colony of these fish over again in SRTC. The zebrafish in particular is really nice. The genome sequence is done. We're getting pretty big now, 1.5 billion base pairs, about 30,000 genes, but they're transparent, particularly as embryos, so they're also really easy to look at, easy to make lots and lots of them, keep them under controlled conditions, etc. Moving up in terms of size, all of my yeast biologists said they're not higher eukaryotes, they're just bigger eukaryotes, relatively speaking. Mice are basically humans as far as we're concerned, with a very few differences. But mammals and other mammals, relatively speaking at a molecular level, are practically identical to each other. And so the vast majority of things that we'll talk about in terms of very specific mammalian biology, with a few exceptions, again, I'll bring them up when we get to them. I mean, looking at mice, because mice you can do genetics on, you can control whom they mate with, and they have a very similar set of genes, and we'll see this in just a second later on. So what do these guys actually look like? Here's the eukaryotic model organisms. Here's yeast up here. No, it doesn't look much like us, but a lot of its regulatory mechanisms, the DNA, how the DNA is packaged, how DNA is replicated, how it's transcribed, etc., is extremely similar in yeast as it is to us. Here's C. elegans. This, I believe, is one of the mature hermaphrodites. They have hermaphrodites and males. And literally each of these cells down here, you can say, this cell came from the leftward division of the egg after it was fertilized. And the cell lineages are really amazing here. Drosophila, down here at the bottom, the classic experiment was the white-eyed Drosophila. I'm still looking for white-eyed Drosophila in my compost. You didn't see any. But these are really nice from a mutagenic point of view. Here's a Arabidopsis up here in the corner. Number of people use Xenopus. These are these African clawed frogs. These are wonderful in terms of looking at developmental biology because they got really big cells, particularly really big embryos. Genetics are a bit of a pain in those organisms. And then zebrafish down at the bottom here, kind of pretty. Unfortunately, you can't visit the lab where we have all of them being reared. But most importantly, if you're looking at embryos, they're completely transparent, so it's easy to see what's going on in any of those particular embryos. What do I mean when I say that mice are basically humans or humans are basically mice? This is just the example that they use in the textbook, which is a mutation in a tyrosine kinase gene. This poor baby actually has exactly the same mutation as the mouse next to it, has exactly the same phenotype in terms of a coloration phenotype. There are other phenotypes that go with that as well. But that basically gives you an idea how similar one particular gene and the mutation and the phenotype of that one particular gene is going to end up being. If you look at the, let me move this. Go back in time, and this is now just looking at animals. Relative to mice, it's only been, millions of years. About 40 million years of evolution that's happened between the last common ancestors of mice and humans, and 84% amino acid identity in the vast majority of proteins. And when we look at protein structures later on Friday and then on Monday, probably next week too, anything that's above 30% identical at an amino acid level is probably going to be pretty darn identical in terms of function. So any of these animals, going back here even to lampreys, 35%, again, homologous proteins, similar to each other. That's what the actual, you're likely to have a very, very similar function. So pretty much all of these, lampreys vertebrates, cartilaginous fish. I'm not an ichthyologist. But certainly, these are all vertebrates here. So they're all very, very similar to each other at a molecular level. Physiology, everything else, fascinating and different. We're not going to talk about that for the rest of this class. So any more questions having to do with the stuff we talked about on Monday? Central dogma, DNA goes to RNA goes to protein. Genomes, what's in a genome? Again, we'll talk much more about these as we move along. Gene families, again, they're homologs, which can be paralogs or orthologs relative to each other. And why model organisms? Basically, is just they're easier to work with than most of the other organisms. The other thing that I wanted to mention is that there are usually good tools for these different organisms. For a long time, there weren't very many whole genome sequences that were available. And so having a whole genome sequence available for the organism you're working with makes everything else you do that much easier. Okay, questions on this stuff before we move on to everything you need to know about chemistry, at least as far as this course is concerned? Okay, so we'll talk a little bit about the atoms in biology. This is great because we don't have to care about very many of them. Basically, it's organic chemistry. Everything in biology, well, not everything in biology, but most of the stuff we'll talk about in this class has to do with weak interactions. And those are going to be your van der Waals interactions, your hydrogen bond interactions, and your ionic interactions. All of biology as we know it is in an aqueous solution. So water, That's also one of the reasons that NASA has been looking for water and other planets, because everything we know about biology has to do with the chemistry that makes biology possible. All of the chemistry that we know that makes biology possible is aqueous chemistry. So that's one of the reasons that NASA has been looking for it. And one of the reasons that aqueous chemistry is so useful is because water is just an absolutely incredible molecule. And we'll talk more about that as we move on here. Yes, there are also strong interactions, and basically the whole idea with strong interactions is going to be covalent bonds. And as far as we're concerned, we're mostly going to be talking about monomeric molecules coming together to make polymers or polymeric molecules. Talk about amino acids becoming proteins, nucleic acids, individual nucleotides becoming nucleic acids. And that's really the majority of the strong interactions we're going to be talking about, having to do with bond energy. Talk a little bit today about activation energy, the high energy bonds, everyone's favorite being ATP, and how biology can actually work, because I forget, one of my colleagues always used to say, all that death is equilibrium. So we're not in equilibrium right now. None of life is in equilibrium, and how life can be not at equilibrium is important. And then, given where we're at today, we probably won't get to protein structure today. So what atoms do we care about? Basically, the ones that have the red boxes around them. The 90+% of the atoms that are present in us and all other living organisms are going to be hydrogen, carbon, nitrogen, and oxygen. And all of the particularly covalent interactions that we're going to talk about are all going to be between those. The other ones which are also important, and we're going to spend more time talking about, are phosphorus and sulfur. And these are present in those nucleotides and proteins that we'll talk about a little bit later on. Any of you who've decided to follow me on Twitter may have noticed that I posted a different form of or a link again to the different form of the periodic table that my great uncle came up with before he went insane. So, if you want to follow me on Twitter, which is absolutely not required. So in terms of chemical bonds, I don't like ionic bonds. Again, I could argue probably a lot with our chemistry colleagues about this. But when we talk about bonds, it's really going to be covalent bonds and then hydrogen quote-unquote bonds. But the important thing here is that with a covalent bond, you're literally sharing electrons between atoms. And so that's what we have over here on, it's the left-hand side for you guys. And the ionic interactions, you have literally a transfer of an electron from one atom to another atom. These are really the two major kinds of things that we'll be looking at. We'll see stably charged, particularly amino acid side chains, but also parts of nucleotides. And then we'll have things which have covalent bonds relative to each other. Important for us is really this relative amount of energy that you have for any of these different kinds of interactions. This, again, up the top here is a log scale going from average thermal motions, particularly it's going to be about 37 degrees Celsius, going all the way out to complete glucose oxidation in terms of the energy that you have. Average thermal motion, if you've got less energy than the water molecules banging against each other, nothing is going to be stable. We'd all just sort of poof apart into the rest of the universe. So we have to have something which has more energy than that. Non-covalent bonds are actually, again, on a log scale, not that much more than average thermal motion. The main important thing here, and we'll talk much more about this, is that if you have lots and lots of these that add together, then it gives you enough stability, but also gives you enough flexibility, because if everything were solid, we couldn't really do anything. We couldn't be dynamic. We couldn't move around at all. So having that kind of flexibility is not just important from a physiology point of view, but it's also really important from a molecular point of view. So it's really all about these weak interactions relative to each other. Covalent bonds are much stronger. That's going to be here, about 350 kilojoules per mole for breaking one of these bonds. You do need to have some bonds that form and break, but for the most part, again, the stuff we'll be talking about is all about these weak interactions. And those weak interactions are going to be the van der Waals interactions, we'll talk about in just a second, hydrogen bonds, interactions with each other, and ionic interactions. Again, all of these, half a kilojoule per mole, 4 kilojoule per mole, 12 kilojoules per mole. No, I don't expect you to remember those exact numbers. I do expect you to know that there are a lot less than covalent bond interactions with each other. And in some cases, actually less than average thermal energy kinds of interactions here. So before we talk about these particular interactions, most important is now to talk about chemical polarity as opposed to molecular biology polarity. Molecular biology polarity is what? Front to back, 5' to 3', N terminus to C terminus. Here, chemical polarity is separation of charge. So if you have a polar molecule, the classic polar molecule is water. It has a separation of charge. This end is more negative. This end is more positive. If you have a polar molecule, it's going to like to interact with other polar molecules, particularly water, because we're in an aqueous environment and there's way more water than there is of any of the other molecules. So polar molecules are going to be hydrophilic. They like water. Non-polar molecules, like oxygen down here at the bottom, that does not have a separation of charge, those are going to be hydrophobic. They do not like water, and they're not going to be interacting with water. If they don't like water and we live in an aqueous environment, what are they going to be interacting with? Other hydrophobic molecules. And we'll see that again in just a second here. So what are some of these interactions? Hydrogen bonds, quote unquote. Basically, this is where you have the hydrogen atom, which is being pulled in two different directions by an otherwise electronegative atom. So you can have this be a hydrogen bond between nitrogen and oxygen, as we have here, and we'll see this in many cases when we talk about proteins, when we talk about nucleic acids. But you can also have hydrogen bonds between two oxygens or between two nitrogens. Just depends on where they are, and very importantly, this interaction is a directional interaction. If you don't have the hydrogen in the line between these two electronegative atoms, you don't have this particular bonding interaction. So the orientation of these is really, really important. And when we talk about Watson and Crick structure, that was the big deal. You had the hydrogen bonds in the middle because they were all pointing at each other. And you have to be pointing straight at each other. All these things have to be linear with each other. Otherwise, you don't get any extra energy from those kinds of interactions. The other short distance interactions are the van der Waals interactions. These are actually the shortest interactions. I won't get into all of the induced dipoles, but again, it's a charge interaction that happens, but it only happens when atoms are very, very close to each other. And it turns out that there is a optimal interaction distance between these. Again, I don't expect you to remember the details here, but what you have is a particular distance between the centers of atoms. Again, it's relatively short. It's on the order of about 4 angstroms, that has the optimal attraction, again, due to these inducing dipoles that are happening. If you get too much closer, this actually becomes repulsion. You get further away, that interaction becomes weaker and weaker and weaker. And in fact, very, very rapidly. It decreases with the 6th power. And so what many people do, you've probably seen actually even in the opening image in D2L when you go to the website, I actually have what they call space-filling model of DNA. And that's the van der Waals radii of each of the atoms that are present in DNA. And the van der Waals radius is going to be exactly at this point right here, which is your optimal interaction point. Very, very weak for each of the individual interactions, but if you have lots and lots of them together, it ends up being rather strong. And this is now not a directional thing, not like what you have with hydrogen bonds. It's literally proximity. And with that proximity, then you can get those. But it's got to be really a good fit, because if it's not a good fit, then you end up in these areas with either not attractive, or if you're trying to push things in too close, these end up being repulsive. Potentially one of the most important of these interactions are hydrophobic interactions. Now, I like to think of hydrophobic interactions as actually a whole bunch of hydrogen bonds that form between water molecules. So I mentioned you can have hydrogen bonds forming between oxygen. So you've got oxygen, part of your polar water molecule. There's a slightly negative charge. You can line up your hydrogen atom, which has a slightly positive charge, nice linear interaction. This gives you a hydrogen bond. One of the things that happens when you have a hydrophobic interaction is you have these molecules that don't interact with water, the gray ones down here at the bottom. Those then are going to come together, but most of the reason energetically that they come together has to do with the fact that when they come together, the water can actually form more hydrogen bonds. So the closer these hydrophobic molecules are to each other, the water, which is everywhere, we're all in aqueous environments, is going to be able to form more hydrogen bonds. So it's actually the energy that you get of the hydrophobic interactions is really maximizing water's hydrogen bonding with other water molecules. So add all these things together, you get what we see in molecular biology. So proteins interacting with each other, proteins interacting with nucleic acids interacting with each other. It's a whole bunch of different weak interactions that then give you specificity, i.e., again, with hydrogen bonds, it's got to line up properly. With the van der Waals interactions, they've got to be really close. If they're not close, they don't fit with each other, you're not going to get good interactions. But these are also relatively weak interactions, so things can come apart and go back together. If you didn't have that dynamics, we'd be equilibrium, we'd be dead. So that process, the whole process of dynamics. The one thing that I haven't talked about, you probably say, Dr. Stedman, didn't you talk about ionic interactions? Well, what kind of ionic interactions do we care about in molecular biology? Really has to do with acids and bases. And the main acids and bases we're going to talk about, nucleic acids and the acidic and basic amino acid side chains. So what do we say when we talk about acids? Really, acids are molecules that can give up hydrogen atoms. So here we have acetic acid, which loses a hydrogen atom, becomes an acetate ion, stably negatively charged. Whenever this happens, that hydrogen's got to go somewhere. In an aqueous environment, it's almost always going to go to water. And it's this balance of hydronium ion versus hydroxyl ion. This is what gives you the pH of your particular system. We're going to be talking about biological systems. This is very close to neutral pH. Things will vary a little bit based on exactly where you happen to be in a macromolecule or in the cell. So what molecules are we going to talk about? I should have put some big red lines through some of these because we're not going to talk about fats, lipids, and membranes. We're not going to talk about polysaccharides. We are going to talk a whole bunch about proteins and nucleic acids. So What we're mostly going to talk about are, at least for the chemistry point of view, we'll talk about adenosine triphosphate. Shouldn't be anything new for any of you. And then how these things fit together. Adenosine triphosphate is really our energy source. Nucleic acids are the polymers of the nucleotides. Sugars are polymers of the sugars, or to say polysaccharides. And then lipids are going to be multiple fatty acids. But that's basically all we're going to talk about in terms of sugars and lipids. Most of our time we're going to talk about, again, proteins and nucleic acids. Why are we spending our time talking about proteins and nucleic acids? Because if you ignore the water, which is what most cells are made of, 70%, and this particular example is for bacteria, but it's true for pretty much all cells, with some very slight exceptions. Of course, it's biology, there always can be some exceptions. But if we look at the 30%, which is not water inside any given cell, it's mostly proteins and nucleic acids. Lots and lots of RNA. Most of that RNA actually turns out to be ribosomes. And then pretty small amounts of DNA, lots and lots and lots of protein. So these macromolecules, how do you get them? Basically two kinds of reactions that we're going to talk about in this class, all having to do with our favorite molecule, water. So two kinds of reactions. You have condensation reactions and hydrolysis reactions. Condensation reactions, you lose water. Hydrolysis reactions, you add water. Condensation, bringing molecules together. Monomers going to polymers. Hydrolysis, taking those monomers apart, or just breaking your covalent bonds. So this example I have at the bottom I don't like terribly much because you end up with a double helix, which you actually don't normally have. But the idea here is you've got small precursors, in this case monomers, which are going to come together, and usually this should just be one strand as a polymer here. Condensation, which is the removal of water, is an energetically unfavorable reaction. Hydrolysis is an energetically favorable reaction. And most of this has to do with entropy. If you think about entropy, we're going to maximize entropy, maximize disorder in the universe. Taking two things apart is clearly increasing entropy. Putting two things together is going to be decreasing entropy. So condensation, pulling two things together, energetically unfavorable. Pulling things apart, energetically favorable. Well, how can you do this? And again, we're not at equilibrium. How does life end up doing this? The way that life does this, the joys of biochemistry, are by taking these reactions which are energetically unfavorable, which are going to be what? Mostly with water? Condensation reactions, and coupling them to hydrolysis reactions, which are going to be energetically favorable. And you have to have your favorable reaction have more energy than your unfavorable reaction. Because overall, you have to have a system where you can't actually gain any energy. It's always going to be a little bit of energy loss. Again, increasing entropy, increasing heat. So you have to have these things that are coupled to each other. And probably my favorite image in this whole textbook is the next one, because I know too much about this particular case. We have a spontaneous reaction, which leads to disorder, and organized effort requiring energy input to get it back to an ordered situation. So this is really what biology is about in biochemistry. So let's talk about how we get that to happen. First, actually, these are the slides from last year rather than from this year, but be that as it may. Where does energy come from that we use on the planet? It comes from that thing that we never see here in Oregon, not this time of year. Photosynthesis. It's the fusion energy in the sun, which then generates carbon-carbon bonds. And that's here, this process of photosynthesis. which actually will take our water, undergo condensation reactions, make lots of nice organic molecules, and then these organic molecules will hydrolyze and generate water, giving us chemical bond energy. So how do we do enzymes? And these enzymes, all that an enzyme does doesn't change an equilibrium. Again, equilibrium is just the relative energies of your products and your substrates. All that enzymes do, or for that matter just any catalysis does, is it makes the reaction faster and easier to actually happen. So if you think about this from a free energy point of view, and hopefully this is stuff you've all seen again in general chemistry, partly why I'm going through this so quickly, is reactants going to products. X is energetically favorable, but Y doesn't spontaneously go to X in the absence of getting a little bit more of extra energy so that it can progress across this activation energy barrier. All that enzymes do is reduce this activation energy barrier. And enzymes in general, what are they doing? They're actually binding to substrates and bringing them close together. And then you can have the reaction take place. It'll just happen a lot faster when you bring these two things together. If it's an energetically unfavorable reaction, it's never going to happen, even if you bring them right next to each other. It still has to be energetically favorable, but you want to make sure that you can speed up the rates of those reactions through interactions with enzymes. So this is shown schematically. Here, energetically favorable reaction, again, going from Y to X. Here's, and usually what's this going to be from our point of view? That's going to be a hydrolysis reaction. You need to have a condensation reaction down here at the bottom. Energetically, this has to be a larger amplitude than this reaction. Here we have our reaction going large change in free energy. Free energy here is just delta G, coupled to positive delta G. And how that is done in biological systems almost always is going to be done by some kind of so-called activated intermediate. So you take one reaction and you couple it to a different reaction, again, very often in the presence of adenosine triphosphate. Here you have glutamic acid. What you need to do is put an ammonium group on it, again, making a covalent bond. In order to make a covalent bond, you need to break some covalent bonds. The breaking of the covalent bonds, the way you couple this breaking of covalent bonds, getting energy, to using that energy to make this bond is through a high energy intermediate. Almost always is going to be a phosphorus group here. And if we want to get into all of the organic chemistry, really good leaving groups in terms of that process. Activated intermediates are how you can get biology and biochemistry to work, going mostly from monomers to polymers, which is what we have here. Again, hopefully this is all review for people. This is why I'm going through it quickly and got questions about it. Slow me down. High energy bonds in terms of polymerization. It's true for polysaccharides. We're not going to talk about that. We're going to talk about how it works for proteins and how it works for nucleic acids. For nucleic acids, turns out nucleic acids are hard to polymerize because the phosphodiester bonds, which are being formed, they're relatively high energy bonds. So it turns out that just one bond hydrolysis is insufficient to get that condensation to take place. You actually have to hydrolyze two bonds, and that process that we have for polymerization of nucleic acids first is hydrolysis of the what we call the gamma phosphate, and then also the beta phosphate. So the two bonds that get broken here from ATP will then, and also all the other nucleotide triphosphates, that and only the generation of both of these is going to be sufficient energy to move forward. So I see the other classes trying to get in here. I will try and upload clicker scores probably by next week. Got any clicker questions, please hold off on those. I'll see you on Friday.